Hello everyone, my name is Emeraldo Souza and today I'll be presenting on the regulation of glycolysis and gluconeogenesis with a particular emphasis on the role of Aldolase and transcriptional control. This presentation is part of the CHM 530 Concepts of Biochemistry course under the guidance of Professor Dr. Hector Rivera Jaques. The goal of today's lecture is to move beyond memorization of pathways and instead focus on understanding how these pathways are regulated and integrated at both the enzymatic and genetic levels. Glycolysis and gluconeogenesis are often taught as opposite pathways, but in reality, they function as a coordinated system that allows the body to adapt to changing nutritional states. By the end of this presentation, you should have a clearer understanding of how these pathways are controlled and why enzymes such as Aldolase play a central role in metabolic regulation. As we begin, let's take a moment to review the learning objectives for today. First, we will review the fundamental steps and regulatory principles of glycolysis and gluconeogenesis. This will ensure that everyone has a solid biochemical foundation before we move into more advanced concepts. Second, we will examine aldolase as an enzyme that participates in both pathways. This is important because aldolase illustrates how a single enzyme can function in opposing metabolic directions depending on cellular conditions. Third, we will compare the different aldolase isoforms and explain why specific tissues express different variants of the enzyme. Finally, we will explore how aldolase B expression is regulated at the transcriptional level by hepatocyte nuclear factors C, EBP, and D, BP. These objectives reflect the progression from basic metabolism to molecular regulation. As we move on our lecture, let's talk about the glucose breakdown pathway. Glycolysis is the central pathway for glucose breakdown and occurs in the cytosol of all cells. It is an oxygen-independent process, which means it can function under both aerobic and anaerobic conditions. During glycolysis, 1,6-carbon molecule of glucose is converted into 2,3-carbon molecules of pyruvate. From an energetic perspective, glycolysis produces a net gain of 2 ATP molecules and 2 NADH molecules per glucose. Although this ATP yield is relatively modest, glycolysis is extremely important because it provides rapid energy, particularly in tissues such as muscle and red blood cells. In addition, glycolysis generates metabolic intermediates that serve as building blocks for other pathways, including the citric acid cycle and biosynthetic processes. Several steps in glycolysis are tightly regulated, and this regulation occurs in three key enzymatic control points. The first one is hexokinase or glucokinase in the liver. These three enzymes catalyze the phosphorylation of glucose to glucose 6-phosphate, effectively trapping glucose inside the cell. Hexokinase is found in most tissues and has a high affinity for glucose, while glucokinase which is expressed in the liver, has a lower affinity and functions as a glucose sensor. The second and most important regulatory enzyme is phosphofructokinase 1, commonly referred to as PFK1. PFK1 catalyzes the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate and represents the rate-limiting step of glycolysis. Because of this, PFK1 serves as the primary control point for glycolic flux and integrates signals related to the cell's energy status. The final major regulatory enzyme is pyruvate kinase, which catalyzes the last step of glycolysis, converting phosphoenolpyruvate to pyruvate while generating ATP. This step helps commit carbon atoms to downstream pathways such as the citric acid cycle or fermentation, depending on the cellular conditions. Together, these three enzymes, hexokinase or glucokinase, phosphofructokinase 1, and pyruvate kinase, allows glycolysis to respond dynamically to cellular and hormonal signals, ensuring that glucose metabolism is matched to the energy needs of the cell. Let's now talk about the regulation of glycolysis. Glycolysis is regulated at multiple levels to ensure that glucose metabolism is matched to the energy needs of the cell and of the organism as a whole. These regulatory mechanisms include energy charge, allostatic control, hormonal signals, and tissue specificity. First, regulation occurs through the cellular energy charge. When ATP levels are high, this indicates that the cell has sufficient energy and glycolysis is downregulated. In contrast, when AMP levels rise, this signals low energy availability, which activates glycolysis. This regulation is mediated primarily through phosphofructokinase 1 or PFK1, which responds directly to ATP and AMP concentrations. Second, allostatic regulation further fine-tunes glycolysis. Metabolites such as citrate inhibit PFK1, linking glycolysis to the citric acid cycle, while fructose 2,6-bisphosphate is a powerful activator of PFK1. Fructose 2,6-bisphosphate serves as a key signaling molecule that reflects hormonal status, particularly insulin and glucagon levels. Third, hormonal regulation allows glycolysis to be coordinated at the whole body level. In the fed state, insulin promotes glycolysis, especially in the liver, by increasing fructose 2,6-bisphosphate levels and activating glycolytic enzymes. In contrast, during fasting, glucagon inhibits glycolysis in the liver while promoting gluconeogenesis. This hormonal control ensures that blood glucose levels are maintained within a narrow physiological range. Finally, and very importantly, glycolysis is regulated through tissue specificity, meaning that different tissues respond differently to glucose and hormonal signals. For example, liver and muscle cells have distinct metabolic roles. The liver functions as a regulator of blood glucose levels. When glucose is abundant, the liver increases glycolysis and glycogen synthesis. During fasting, the liver suppresses glycolysis and instead produces glucose through gluconeogenesis to maintain blood glucose levels. In contrast, skeletal muscle uses glucose primarily for its own energy needs. Muscle cells increase glycolysis during activity to generate ATP, but do not release glucose back into the bloodstream. Unlike the liver, muscle cells do not respond to glucagon in the same way and lack key gluconeogenic enzymes. This tissue-specific regulation ensures that glycolysis supports both local energy demands, such as muscle contraction, and systemic glucose homeostasis, which is primarily controlled by the liver. Moving on, let's now talk about the glucose synthesis from non-carbohydrates. Gluconeogenesis is the metabolic pathway responsible for the synthesis of glucose from non-carbohydrate sources. This pathway plays a critical role during periods when dietary glucose is unavailable, such as during fasting, prolonged exercise, or starvation. Gluconeogenesis occurs primarily in the liver, which accounts for approximately 90% of total activity, and to a lesser extent in the kidney cortex, which contributes about 10%. This tissue distribution reflects the liver's central role in maintaining blood glucose levels. Because several steps of glycolysis are irreversible, gluconeogenesis cannot simply run glycolysis backward. Instead, it uses strategic bypass reactions which involve different enzymes that circumvent the irreversible glycolytic steps. This separation allows glyconeogenesis to be independently regulated. The key precursors for gluconeogenesis include lactate produced by muscles, amino acids derived from protein breakdown, and glycerol released from fat metabolism. Together, these substrates allow the liver to synthesize glucose and sustain glucose-dependent tissues. As we continue our presentation, let's talk about the gluconeogenesis reciprocal control. Gluconeogenesis is regulated in a reciprocal manner with glycolysis. This means that when glycolysis is active, gluconeogenesis is suppressed. And when gluconeogenesis is active, then glycolysis is downregulated. This reciprocal control prevents futile cycling and conserves energy. Hormonal regulation plays a central role in this process. During fasting, glucagon and cortisol activate the transcription of key gluconeogenic enzymes, promoting glucose production by the liver. Gluconeogenesis is also energetically expensive, requiring the consumption of 6 ATP equivalents per molecule of glucose synthesized. Because of this high energy cost, gluconeogenesis occurs only when sufficient energy is available. And then, mitochondria are essential for gluconeogenesis. In particular, the conversion of pyruvate to oxaloacetate within mitochondria initiates the pathway, highlighting the close integration between mitochondrial and cytosolic metabolism. Now let's talk about the aldolase, the glycolytic workhorse. Aldolase is a central enzyme in glycolysis and is often referred to as the glycolytic workhorse. It catalyzes the cleavage of fructose 1,6-bisphosphate into 2,3-carbon molecules. These products are glycerol-dehyde 3-phosphate and dihydroxyacetone phosphate. Although these molecules are different, they are readily interconverted, allowing both carbons to continue through glycolysis. This reaction represents a critical junction in the pathway because it links the energy investment phase of glycolysis to the energy payoff phase. Without aldolase activity, glycolysis could not proceed to the ADP generating steps that follow. Now let's talk about the aldolase in gluconeogenesis. In glycolysis, aldolase splits fructose 1,6-bisphosphate. In gluconeogenesis, aldolase catalyzes the reverse reaction, condensing triose phosphates to reform fructose 1,6-bisphosphate. Importantly, this reaction is carried out by the same enzyme. Aldolase is, therefore, a bidirectional catalyst capable of functioning in both metabolic directions. The direction of the reaction is determined not by the enzyme itself, but by the metabolic context of the cell. Changes in substrate concentrations and pathway flux determine whether aldolase supports glycolysis or gluconeogenesis. Now let's talk about the isoform diversity, the three aldolase variants. Humans express three aldolase isoforms known as aldolase A, aldolase B, and aldolase C. Aldolase A is predominant in muscle and erythrocytes and optimized by high glycolytic flux, supporting rapid ADP generation. Aldolase B is expressed primarily in the liver, kidney, and small intestine. This isoform handles both glucose and fructose metabolism, reflecting the metabolic role of these tissues. Aldolase C is found in the brain and neural tissue, where it supports the unique energy demands of neurons. Each isoform is encoded by a distinct gene located on separate chromosomes, allowing precise tissue-specific expression. But why multiple isoforms matter? The presence of multiple aldolase isoforms allows tissues to meet specific metabolic demands. Each isoform exhibits different kinetic properties and substrate preferences. Aldolase B has a unique specialization. It efficiently cleaves fructose 1-phosphate, a critical intermediate in dietary fructose metabolism. Clinically, mutations in the aldolase B gene result in hereditary fructose intolerance, a potentially fatal condition characterized by severe hypoglycemia and liver dysfunction. This illustrates why isoform specificity is essential for metabolic health. As we move on, let's talk about the transcriptional regulation of aldolase B. Aldolase B expression is regulated at the transcriptional level and is largely restricted to the liver. This liver-specific expression is controlled by hepatocyte-enriched transcription factors, ensuring that aldolase B is produced only in appropriate tissues. Aldolase B expression is also nutritionally responsive. Gene expression increases in the fed state and decreases during fasting, allowing metabolism to adapt to nutrient availability. Finally, Regulation occurs at the level of transcription initiation, demonstrating that metabolic control extends beyond enzyme activity to gene expression. Now let's talk about the hepatocyte nuclear factors, or HNF1 and HNF3. So we're going to be talking about the role of the hepatocyte nuclear factors 1 and 3. And they play a central and foundational role in regulating aldolase B expression and, more broadly, in establishing liver-specific gene expression. These transcription factors ensure that metabolic genes are expressed in the correct tissue and at the appropriate level. HNF1, or hepatocyte nuclear factor 1, binds directly to specific DNA sequences located in the promoter region of the aldolase B gene. This binding is essential for basal transcription, meaning that without HNF1, aldolase B expression is dramatically reduced or absent. HNF1, therefore, acts as a gatekeeper for liver-specific transcription. Now, HNF3, also known as FOXA, plays a complementary but distinct role. HNF3 is considered a pioneer transcription factor, meaning it can bind to condensed chromatin and help open the DNA structure. By doing so, HNF3 allows other transcription factors, including HNF1, to access the promoter region more effectively. Together, HNF1 and HNF3 establish a transcriptionally active chromatin environment in hepatocytes. This cooperative interaction ensures that aldolase B is expressed selectively in liver cells and supports the liver's specialized role in carbohydrate and fructose metabolism. As we continue our lecture, let's talk about the role of CEBP and DBP. In addition to hepatocyte nuclear factors, aldolase B expression is regulated by transcription factors that integrate nutritional, hormonal, and temporal signals. These important examples are CEBP and DBP. The CEBP family, or CCAAT enhancer binding proteins, plays a major role in regulating genes involving in energy metabolism, inflammation, and differentiation. In the liver, CEBP activity increases during the fed state when carbohydrate availability is high. This allows CEBP to enhance aldolase B transcription, supporting glycolysis and fructose metabolism. CEBP acts as a metabolic sensor, translating hormonal signals such as insulin into changes in gene expression. This ensures that aldolase B levels rise when glucose and fructose are abundant. DBP, or D-box binding protein, introduces an additional layer of regulation by linking gene expression to the circadian clock. DBP expression oscillates throughout the day, peaking during periods of active feeding. By regulating aldolase B expression in a time-dependent manner, DBP aligns hepatic glucose metabolism with daily feeding fasting cycles. As we're getting to the end of our lecture, our presentation today, let's now talk about the clinical application of what we have discussed so far. or presented so far. And this means the physiological integration and disease. The regulatory mechanisms discussed so far provide the body with metabolic flexibility, allowing it to adapt efficiently to changes in nutrient availability. This flexibility is especially important during transitions between fed and fasted states. The liver functions as a metabolic hub, coordinating glucose uptake, storage, production, and release. During feeding, hepatic glycolysis and glycogen synthesis dominate. During fasting, gluconeogenesis becomes the primary pathway for maintaining blood glucose levels. When these regulatory systems fail, metabolic disease can result. Excessive gluconeogenesis is a hallmark of type 2 diabetes, contributing to chronic hyperglycemia. Conversely, defects in Aldolase B impair fructose metabolism and lead to hereditary fructose intolerance, a condition characterized by hypoglycemia, liver damage, and gastrointestinal symptoms. Understanding these pathways is clinically important because metabolic enzymes and transcription factors represent potential therapeutic targets. Modulating gluconeogenic gene expression or autolase activity may offer strategies for treating metabolic disorders. As we're getting to the end of our presentation, our lecture today, let's just summarize what we have talked or discussed so far. The concepts presented in this lecture are grounded in foundational biochemistry research and supported by authoritative academic sources. Classical biochemistry textbooks provide detailed descriptions of glycolysis and gluconeogenesis, while peer-reviewed literature expands on transcriptional regulation and nutrient-responsive gene expression. Taken together, these sources illustrate how metabolism is regulated at multiple levels, from enzyme kinetics to chromatin accessibility and transcriptional control. Aldolase serves as a powerful example of how a single enzyme can participate in opposing pathways while being regulated in a tissue-specific and time-dependent manner. In summary, glycolysis and gluconeogenesis are not isolated pathways, but components of an integrated metabolic network. Transcription factors such as HNF1, HNF3, CEBP, and DBP ensure that autolase B expression is precisely controlled in the liver, allowing the body to maintain glucose homeostasis. And I want to thank you, all of you, for your attention, and please refer to the listed references for further reading. Thank you again. Until next time.